I realized she wasn't going to live that long.
But to be able to clone their mother in the future, they would have to get a tissue sample from her and have it frozen, and they couldn't find a scientist willing to do the job.
It had come down to where my brother and I were going to do it after my mom passed away. And so we contacted someone who told us how to do it. You could either do it from like the upper arm or from her leg, from her lower leg. And you could actually do it with, like, you know, scissors. Everything would have to be sterilized. You use gloves.
Fortunately, they didn't have to, and a doctor agreed to perform the vital operation properly.
I just went in just by myself with a doctor, and we actually had 6 vials. Then he took some tissue samples, you know, he cut them up and put them in the samples and then sealed them. We actually then, the next day, we FedExed them to this laboratory.
Now 6 pieces of Al's mother are frozen in a vat somewhere in America, waiting for the era of human cloning to begin. But until recently, many thought that era would never come. For over 30 years, researchers had been trying to clone animals from adult cells. It had never worked. By the 1980s, many leading scientists were saying it was impossible.
If you asked some geneticist 25 years ago whether we would be doing the things that we're doing today, even whether we would know the sequence of the entire human genome, they would have said, no way that's going to happen in that period of time.
But in February 1997, the age of cloning began, with a sheep called Dolly. So far, cloning has been successful in sheep, cattle, pigs, goats and mice. But the scientists doing it were never aiming to make human babies. Instead, they see cloning as a new kind of farming, a way to create animals genetically engineered for our benefit. In the next few years, this cattle fair might be trading animals which make human antibodies or medicines in their milk. This will be possible because of cloning. Genetically engineer one animal and you'll be able to clone a whole herd. And away from the hype about cloned babies, mainstream scientists do also see good reason for cloning from human cells. They're hoping to treat incurable conditions like Parkinson's or heart disease by growing new brain or heart cells cloned from the patient. But behind all the optimism lies a stark reality. At the Roslin Institute, the lab which made history with Dolly, scientists have found their success very hard to repeat. For every 100 cells injected into eggs, typically only 2 or 3 clones will be born alive. Most will either fail to start growing into an embryo at all, or fail to implant in the womb. And in the few cases where pregnancies begin, many of the cloned animals die before they even make it to birth.
It can happen at any time during the development of pregnancy. Sometimes my colleagues would see the placenta going wrong. Sometimes things would look perfectly normal. You'd be expecting a live birth within a few days, and the lamb would die, the fetus would die, because of some abnormality that you couldn't detect by ultrasound.
Then there was the strange case of a cloned lamb, which began life seeming perfectly normal.
Well, we were very excited by the fact that it was born naturally and was essentially very healthy, except for one thing, which was that it panted all the time, just the same way that you and I would if we'd been running for 100 yards, but all of the time, night and day. And obviously my veterinary colleagues took advice from other vets and from the children's hospital and tried any of the treatments that were recommended, but unfortunately none of them were able to correct the condition.
And it wasn't just the Roslin lambs. Around the world, some clones were dying of bizarre, inexplicable conditions.
In cloned animals of all the different species, there have been an enormous variety of different abnormalities. In the kidneys, the livers, the hearts, the blood vessels, the skin, the musculature of the body wall.
Heart defects, limb abnormalities and facial abnormalities.
Newborns have been born with their insides on the outside. We know that they have immunological problems, they have pulmonary problems, they have cardiac
and then
problems.
within hours of birth they tend to die.
The musculature of the body wall, the brain, the face.
Some of them
I think
are able to breathe, but only breathe if they're on oxygen. Who ever heard of a calf grazing with oxygen on its back?
Scientists had to get to the bottom of the mysterious deaths. They took the organs from clones which had died and prepared tissue samples. Pathologist Susan Rind was one of the first to examine samples from dead clones under the microscope. What she has found is shocking.
These lambs showed serious problems with their breathing, so an obvious place to start looking was in the lung tissue itself to see if there was any problems there. And if we look at a section here from a normal lamb, and what we've got here is a large airway bringing the air down into the lung. And if we can focus on this structure here, which is a blood vessel, and at this same magnification move on to looking at the lung from a cloned lamb, and we can see instantly how different they are from the normal. We have a blood vessel which is massively enlarged, and the size of this is really far beyond what one would normally expect in a normal lung.
The more samples she looked at, the worse the picture became.
This next slide here is too big to even fit on the screen. I mean, this is an even more massively enlarged vessel.
The lambs' hearts couldn't cope with the task of trying to push blood round gigantic vessels 20 times bigger than they should be. These clones had no chance.
It's not possible to pick up these abnormalities by ultrasound and carry out an...
For the Roslin Institute's science director, Harry Griffin, the fate of clones like these should sound a warning to anyone who wants to clone a human.
Told us that this animal was suffering from a major cardiovascular problem that was untreatable. We put this animal down because we didn't want it to suffer any longer. Now, if any of you are tempted with the idea of cloning a human being, and I would ask Dr. Zabas this question, given that sort of defect, what would he do with it?
All those experiments that are done in animals today are obviously done on a hit and miss type basis.
But Dr. Zavos insists that long experience of making human babies makes him a safer pair of hands.
Dr. Zavos has been producing children for the last 25 years, and not one single baby, abnormal baby, was born ever. And we don't intend to have one single child ever be born from such an effort, ever. Lambs are lambs, humans are humans. Dr. Griswold has to realize what the difference is, and that's what the difference is. Thank you.
The difference in this case, of course, is that in the UK and elsewhere we're allowed to experiment on lambs, we're not allowed to experiment on human beings.